Antimicrobial drugs include antibiotics, the semi-synthetics, and the synthetics. In the early 1900s, 1/3 of all children born died by age 5 from infectious diseases. Early pioneers in this field were Alexander Fleming, who discovered penicillin from mold, and Paul Ehrlich, who discovered arsenic would kill trypanosomes. Chemotherapy refers to the use of chemicals to kill pathogens with little or no effect to the patient. Please note this is different than what most people assume regarding chemotherapy being only a treatment for cancer. The name implies using chemicals as therapy. Antibiotics are antimicrobial chemicals which are naturally produced by microorganisms. In 1932, sulfonamide was the very first practical antimicrobial put into widespread use. It was effective in a wide variety of infections. The semi-synthetics are chemically modified antibiotics, and the synthetics are entirely synthetic antimicrobials. The goal of most antimicrobial drugs is to use selective toxicity towards the microorganism but not the person. There are several mechanisms of action used by antimicrobial drugs. One way is to inhibit cell wall synthesis. This avoids human cells because they do not have cell walls. The drugs may also inhibit protein synthesis. In some cases, this can be proteins in microbes that do not exist in humans to reduce toxicity. The cytoplasmic membrane may be disrupted, destroying the osmotic balance of the cell. Some will inhibit non-human metabolic pathways. Other antimicrobials may inhibit nucleic acid synthesis. These tend to have more side effects since both microbes and humans synthesize nucleic acids. Another mechanism is to block recognition or attachment of the microbe to the host. First, we will look at drugs which inhibit cell wall synthesis. This will disrupt the mechanism protecting the cells from the osmotic pressure. The beta-lactams inhibit peptidoglycan synthesis by attaching the enzymes that cross-link the NAM subunits. These are only effective on a growing cell. Examples include the penicillins and cephalosporins. Vancomycin is obtained from Streptomyces orientalis and directly interferes with the alanine-alanine bridges linking NAM in the cell wall. This is only effective on growing cells and is beginning to see more antibiotic resistance. Cycloserine is a semi-synthetic that also interferes with alanine-alanine bridges and is only effective in growing cells. Bacitracin interferes with cell walls by blocking the secretion of NAG and NAM present on the walls of Mycobacterium. Since these cells are slow to divide and only reproducing every 12 to 24 hours, it takes months to years of treatment. Drugs that inhibit protein synthesis can interfere with the formation of structural proteins, cell regulation, metabolism, and the formation of enzymes. The aminoglycosides target the 30S ribosome, which is only in prokaryotes. Some examples include streptomycin, amikacin, tobramycin, gentamicin. Kanamycin changes the shape of the 30S ribosome. The tetracyclines also target the 30S ribosomes and block the tRNA docking site. Some examples are tetracycline, doxycycline, and trimocycline. Chloramphenicol blocks the enzymatic site of the 50S subunit, preventing translation. The macrolides such as clindamycin, erythromycin, and azithromycin bind to the 50S subunit and prevent the ribosome from moving from one codon to the next. Drugs that disrupt the cytoplasmic membrane will incorporate into the membrane and damage its integrity. The polyenes attach to ergosterol and lipids in the fungal membrane. The presence of cholesterol makes the human membrane somewhat susceptible, but most bacteria lack sterols and are resistant. An example is the fungicide amphotericin B. The azoles inhibit ergosterol synthesis. This includes antifungals, miconazole, and ketoconazole. Polymyxin is produced by the Bacillus polymyxa. It targets gram-negative cells and Pseudomonas. Due to the nephrotoxicity, it is generally used topically. With drugs that inhibit metabolic processes, we try to target metabolic processes which are different from the host. The sulfonamides are structural analogs to PABA, or para-amino-benzoic acid, used in nucleotide synthesis. Many organisms convert PABA into THF or tetrahydrofolate, used as a coenzyme in purine and pyrimidine synthesis. Sulfonamides, like trimethoprim, block THF synthesis. This does not target human cells because we get THF from food, not PABA. Amantadine and romantadine act on the phagolysosome to prevent viral uncoating. Drugs that inhibit nucleic acids can block the replication of DNA or transcription. The nucleic acids can block the replication of DNA or transcription found in both prokaryotes and eukaryotes, so they are not usually used for infection and only for cancers. The nucleotide analog is a compound structurally similar to a nucleotide. They are incorporated into the genome and change shape, preventing further replication. These are useful with viruses because the viral DNA polymerase is much more likely to incorporate a non-functional base than the host. The quinolones and fluoroquinolones inhibit DNA gyrase to prevent the uncoiling and coiling of bacterial DNA. For example, ciprofloxacin. Rifampin is a drug that binds more readily to prokaryotic RNA polymerase than in eukaryotes. It is used for Mycobacterium tuberculosis and other slow metabolizing pathogens. Clofazimine binds the DNA of Mycobacterium leprae and prevents normal replication and transcription. Relatively new in the world of antimicrobial agents are drugs that prevent viral attachment. It is blocked by the peptide and sugar analogs of the attachment or receptor proteins. The attachment antagonist artidone is active against some cold and polioviruses. Many of these are still being researched and developed. Several clinical considerations must be taken into account when prescribing an antimicrobial. Availability may vary throughout the world and cost may be prohibitive. They also must be stable enough to be delivered to the patient, and the mode of transmission, such as oral versus injection, may be a concern. The drug must be non-toxic and non-allergenic and have selective toxicity for the parts of the target microbe. Narrow-spectrum drugs work on only a few pathogens, while broad-spectrum drugs are effective against many different pathogens. It is best to attempt treatment with the most narrow-spectrum drug first to minimize potential resistance. Superinfection results when there is development of infections by microbes not affected by the antimicrobial. The sensitive organisms may be killed, leaving less competition and more resources available to the resistant organisms. Reduced normal flora increases susceptibility because there is less competition between the normal flora and the pathogens. Candida albicans is an example of an organism whose growth frequently gets out of hand with reduced competition from the flora. This is commonly seen after taking an antibiotic for something like a urinary tract infection, where frequently females will end up with candida or yeast infections following. There are a few ways to evaluate the efficacy of antimicrobial agents. Diffusion susceptibility tests use small discs of paper containing standardized concentrations of a drug placed on inoculated plates. After incubation, they are examined for a zone of inhibition or a clear area surrounding the disc. When microbes don't grow, the MIC, or minimum inhibitory concentration, is the smallest amount of drug that inhibits the growth of the pathogen. This is used in used dilution tests. The MBC, or minimal bactericidal concentration, is determined from diffusion tests. This is the lowest effective concentration killing subcultures of susceptible organisms. With the bactericidal, it's going to kill the organisms, where inhibitory just stops growth. Antimicrobials may be administered through different routes. Topical or local application is applied directly in external infections. Some agents are tolerated in topical applications, but not internal applications. Oral administration is the simplest technique for internal infections, but the drug concentrations are lower and patient compliance is lower. IM, or intramuscular, is given through a needle and diffuses through the blood in the muscle. The concentrations are lower than through IV, or intravenous, administration. IV can achieve greater concentrations, but they also diminish rapidly as the liver and kidney are going to remove the drug. There is always a concern for safety in using drugs. Certain agents may be toxic to the kidney, liver, or nerves, or may not be safe during pregnancy. Polymyxin and the aminoglycosides are nephrotoxic. Metronidazole or Flagyl may result in a black hairy tongue from hemoglobin breakdown and the minerals accumulating in the papillae of the tongue. Tetracycline can form complexes with the calcium in the teeth, leaving discoloration. There is a risk of anaphylactic shock from allergy. About 0.1% of Americans are allergic to penicillin, and many have milder reactions. Antimicrobials also disrupt the balance of the normal flora, leaving a person at risk of secondary infection from the lack of competition. Candida albicans frequently multiplies with the lack of competition. Another common secondary infection is Clostridium difficile in the colon, causing pseudomembranous colitis. Not all pathogens are equally sensitive to antimicrobials. R plasmids, which carry the genes for resistance, may occur in viruses as well as bacteria. The susceptible strain dies, and when the competition decreases for the resistant strain, they're able to flourish. There are several common mechanisms which convey resistance. The beta-lactamases are enzymes which destroy and deactivate the beta-lactams. Porin proteins may slow or prevent entry of the drug into the cell, and this is seen with penicillins and tetracyclines. There may be genes to alter the receptors for a drug, as seen in the sulfonamides and erythromycin. The cell itself may alter its metabolic chemistry or pump out the drug before it can act. An R plasmid may carry resistance to multiple drugs, especially in the medical environment. Resistance is very common in staph, strep, enterococcus, plasmodium, pseudomonas, and a growing list of others. They may also confer cross-resistance, where it's going to convey resistance to similar drugs, which is seen in the aminoglycosides. aminoglycosides.
Bacteria are becoming resistant to many of the antibiotics in our medical arsenal. One reason for the widespread resistance lies in the indiscriminate use of antibiotics. For example, at the insistence of patients, doctors often prescribe antibiotics even for the common cold. Antibiotics have no effect on viruses, and viruses cause most colds. Antibiotics attack bacteria. There are millions of bacteria in the body. Most are found in the gut and are either harmless or beneficial. Some of these bacteria are beneficial because they make vitamins that the body can use. Also, by competing for space and nutrients, the many species of bacteria prevent each other, as well as other harmful bacteria, from multiplying out of control. What happens to the resident bacteria when a person takes antibiotics? If the person has never taken this particular antibiotic before, then most of the bacteria would probably be susceptible to it. Antibiotics work by a variety of mechanisms. Tetracycline, for example, binds to ribosomes in bacteria and jams the production of proteins. From lack of protein production, a bacterium dies. Most of the bacteria die, but by chance, a few may have mutations that allow them to live in the presence of the antibiotic. Their mutations might cause their ribosomes to have a slightly different structure, which prevents the antibiotic from binding. With these mutations, the bacteria can still make proteins and survive in the presence of the antibiotic. They are resistant. The antibiotic treatment provides an example of natural selection in action. In the presence of antibiotics, susceptible bacteria die, but resistant bacteria have an advantage and multiply. The bacteria reproduce asexually and therefore make identical copies of the original cells. The populations of bacteria evolve, with the proportion of resistant bacteria greatly increased. Once established, resistance is hard to eliminate, and the resistance may spread to different species of bacteria. The easiest way to spreading resistance is when the resistance gene is located on a loop of DNA called a plasmid. We've already seen how resistance can occur when the antibiotic's target, such as a ribosome, changes. However, other antibiotics work by different mechanisms. In another type of resistance, a gene might code for a transport protein that pumps antibiotics out of the bacterial cell, preventing the antibiotics from harming the bacterium. In another type of resistance, a gene might code for an enzyme that breaks apart antibiotic molecules. The ability to resist an antibiotic can be gained by plasmid transfer. In a frightening yet realistic scenario, a relatively harmless bacterium transfers its resistance genes to a highly pathogenic species. What can we do to reduce the incidence of antibiotic resistance in bacteria? This brief list outlines important considerations.
We can slow resistance with some precautions. By using a high enough concentration of the drug to kill sensitive cells, it may inhibit others enough for the body's defenses to detect them. Some antimicrobials may have a synergistic effect when used in combination, but care must be used that they may also have an antagonistic effect, which we do not want. Educating doctors and nurses to limit their use to necessary cases, as well as informing the patient of the need to finish prescriptions and avoid the use of other people's or their pets' prescriptions. Pharmaceutical companies also continue to explore new drugs and new variation of existing drugs. One of the more challenging things in dealing with antibiotic resistance is to educate the patient on the need to follow the instructions.